So I was going to talk today because your topic is SARS coronavirus 2 or research in the times of a pandemic. And I was going to talk about predicting targets of immune responses in SARS-CoV-2. I just heard that you just had a talk from Alessandro Sette, and Alex and I are kind of joined at the hip in this area. And I do mainly the bioinformatics parts, and he does mainly the experimental parts. And I was unfortunately not aware of this talk just happening, so I'm hoping I'm not repeating too much, and I just picked up some other potential topics I can talk about. But we'll see. I will stop this talk every once in a while, and I want to specifically, if you, well, I don't know. Actually, I don't know if we can do like a show of hands or something. How many people have actually listened to the talk from Alex who are here now? So I see 2 hands. So that's not that much. I don't know if that's, how many people have not seen Alex's talk? Okay, so there's 2, 3, okay. So it's not everyone. Okay, so fine. So I should just not be too worried about it. Anyway, so I will be talking about our work on predicting targets of immune responses in SARS-CoV-2. And the work we are doing is at the La Jolla Institute for Immunology. And this is where we are. So many people I've met from UCSD don't even realize we exist. We're right next to Moore's Cancer Center. It's the Thornton Hospital, obviously west of the 5. We are on UCSD campus. We are like a not-for-profit research institute, similar like Scripps or Salk or so. And we are on UCSD campus since 2006. And there's like a collaborative agreement, research agreement, and lots of things are happening back and forth, but still we are an independent institution. So yeah, it can be a little bit confusing. So what I wanted to talk about is a little bit about the background, the biology of adaptive immunity, and then in terms of, just to give you an introduction. And then I was going to talk about how we catalog targets of adaptive immunity in the immune epitope database, how we use that data that we catalog in our database to train machine learning algorithms to predict outcomes for not yet tested entities, not yet tested assays, and how we applied that specifically for immune response targets in SARS-CoV-2. So roughly that's my catalog of things that I want to cover today, and it's not necessarily directly in this order, but I definitely will start with the background, because I'm not sure how many people here are aware of the different aspects to adaptive immunity. So obviously, with SARS-CoV-2 being in the news, people have now necessarily gained some understanding, and most of what people talk about is antibodies. So adaptive immunity in general is the part of the immune system that learns. It adapts to experience with other pathogens and learns from that experience and gets better at fighting a next round of infection. And the key part of that is humoral immunity. And humoral essentially means like these are things you can find in fluids that are less than cells. So that's a crappy explanation. Anyway, so the idea is if you have extracellular microbes, so this could be viruses, bacteria, whatever, they can bind to receptors that are on B lymphocytes, B cells, and those receptors, they're actually membrane-bound receptors that can be secreted. And once they are secreted in the circulations, they're called antibodies. And these antibodies can directly bind to a virus or bacteria or whatever, and target them for destruction. Essentially, the antibody has like a part here that is highly variable, and that can essentially, there's a step like called somatic, somatic mutations as part of maturation of the B cell, where this antibody can essentially evolve to bind better and better to the microbe And then essentially you get these very, very high affinity antibodies binding to the surface of a pathogen and targeted for destruction or inhibited from being able to enter a cell. So, but obviously these antibodies are requiring that, that they need to be binding a microbe that is inaccessible, that is free and circulating in the extracellular environment. And then there's cell-mediated immunity, where you have T cells specifically as part of adaptive cell-mediated immunity that are circulating and are interrogating other cells, such as here macrophages, where the T cell directly interacts with the macrophage and can actually detect if there is a pathogen inside of the macrophage, and then trigger all kinds of immune responses. And all of these parts here always involve a step where the T cell directly interacts with another cell and then detects if something is wrong with that cell, essentially, and target an immune response. And as the question when you look at this picture should be, how does the T cell know that this cell is infected? So, and that question is essentially answered by the MHC class 1 processing pathway. And I will illustrate that here in the, first I illustrate that I draw very poorly. This is supposed to be a mouse, and these are the ears that I added because otherwise it looks like a sperm. And so, okay, if you have a virus that infects its host, the virus goes inside through some membranes and then enters cells. That's the goal of the virus, because in those cells it can replicate and spread out and multiply and infect more and more cells. That's the whole point of a virus, that it can do these things and uses the machinery inside the host to make copies of itself and spread this way, and then obviously hope that, say, the host coughs or so and then spreads the virus to the next host. Now, how can an infected cell be detected? So that is based on the MHC processing pathway here, where you actually have something that happens to everything inside the cell. Every protein that is present inside the cell is subject to degradation. So proteins are broken up into individual peptide fragments. And these peptide fragments, if they're from a virus or from a self-protein, some of them get presented on the outside of the cell surface by MHC molecules. So you have individual peptides symbolizing essentially to the outside, or signaling to the outside what is happening inside the cell. And these peptide fragments, which are typically something like 9 to 10 amino acids in length, can be scanned by T cells, who are then have the T cell receptors, and they're looking for peptide fragments that they have not seen before. And if they detect peptide fragments that they have not seen before, they're concluding that something is going on in the cell that they haven't seen before. And that could be a viral infection. That could be a mutation in the cell that leads to expression of a mutated protein that is not normally there. And all of that is reason for the T cell to trigger an immune response. And that could be that it kills the cell. It could be that it releases cytokines that signal to other cells of the immune system that there might be something problematic here and they should come and look, like macrophages. And also, if this is really a bona fide foreign pathogen or a foreign entity that they're detecting, the T cell can proliferate and form a memory cell population that can, upon further encounter of the same epitope, re-expand and very quickly mount another immune response. So a lot of the work that we're doing in the lab is about identifying what is this piece. If I get a sequence from a virus, which are typically open reading frames, so we get these protein sequences, I want to be able to say, this is the part here, this peptide sequence within this protein is what is likely going to be recognized by the T cell, because the T cell does not see the antivirus. The T cell sees peptide fragments presented of what's inside the cell, presented and bound to MHC class 1 on the cell surface. So to understand which peptides are presented here, if I have a virus that infects the cell, I have to understand what's going on inside the cell. And this pathway here, this is an ancient pathway. So essentially in every cell, all proteins are constant subject to degradation. You can think of it as like a constant reboot. The cell doesn't want to just accumulate proteins. It constantly essentially destroys everything that's in there, constant turnover, and then produces the new proteins. And this goes on and on and on. So parts of the newly synthesized proteins, and definitely the older proteins, are marked for degradation by the proteasome. And you have these peptide fragments that can then be, most of them are actually chopped up to individual amino acids that are used for resynthesis. So again, this is kind of like a reboot mechanism of freeing individual amino acids up again, making the same, making a new protein. If you think about it, this is otherwise how cells don't have a mechanism to have a targeted destruction of proteins. They essentially destroy everything continuously to be able to control their protein content. But this mechanism, which is really, really ancient, like archaea have it, has been adapted by the adaptive immune system, where you have then some of these peptides are capable of binding the TAP transporter, which brings them into the ER, where they again can be chopped up some further. But some of these peptides bind to MHC class 1 molecules and are then transported to the cell surface, where they are presented. So you end up with this peptide that was in this protein being presented on the cell surface, where it can be identified or recognized by T cell receptors. And so ultimately, again, the question then in epitope prediction is, given a protein, what is the peptide sequence that a T cell receptor might be able to detect on the cell surface? These steps are things that we've evaluated in many, many different studies. And ultimately, what we find is that all of these are selective. But the most dominantly selective step that I'll talk about today pretty much exclusively is MHC binding. So while essentially 1 out of 7 possible peptides can pass through these steps into the ER, only 1 out of 40 or 50 is able to bind MHC at a reasonable high affinity. And then pretty much like nearly 1 out of 2 peptides on the cell surface can be recognized by a T cell receptor if it actually gets presented efficiently. So I saw some chat entries. I want to see if there's questions already or not. Actually, so yeah, are there questions here? We do have time today. I would love to explain also some of the biological backgrounds, if that is possible. If not, also you can put things in the chat again. Where is my chat?
So as shown in this figure, which by the way is really impressive and very explanatory, but the epitope that was binding to the MHC1 protein, that was binding to it by the ends of that epitope. So does that mean that like the ends are more like are a bigger factor when it comes to binding to that MHC1?
Yeah, so actually they are. So for MHC class 1 molecules, and somehow for some reason I can't find my Zoom stuff back. This is stupid. Okay, maybe here. No. So yeah, so position, you can no longer see my screen, right? Currently? Can you guys still see my screen?
You're just on the desktop. Yeah.
Okay, fine.
You're on the presentation now.
Okay, fine. I lost my some of the controls, but that's okay. Yeah, so MHC class 1 molecules specifically actually bind at the C-terminal residue and at position 2. So pretty much the rest is really unbound. So essentially the specificity of MHC binding, and it's not perfectly drawn. It's really this position 2 and the C terminus most of the time. So those are called also anchor positions. So they kind of like anchor the peptide within the MHC molecule. So the rest is pretty variable. So that means essentially like for a given peptide, even if the anchor was like really exclusive, many different peptides can bind. So that you're going to have like hundreds of thousands of possible peptides that can bind easily for a given MHC1 molecule. I hope that answered the question.
Yeah, besides that, so firstly, does that also, is the same principle applicable when it comes to the binding of the T cell receptor, or?
Yeah, no, so actually, and this is like probably one of my worst pictures. No, so the T cell receptor specifically binds to the things that don't bind MHC. So the T cell receptor actually focuses on the middle part. So kind of the MHC like binds the ends, and then the variable part in the middle is what's most exposed to T cell receptors. Now, both of these are hypervariable. So when I say these kinds of things, those are general rules, but there's plenty of exceptions. But essentially, on average, the T cell receptors are the conformation in which they bind brings them most in contact with like residues 3, 4, 5. That's typically the dominant region in which they contact with. And then there's, as I said, like plenty of examples with other things, but typically the C-terminal residues and the position 2 here are the least accessible because they're like deep in the MHC binding groove. And yeah, all of these are kind of more cartoons. There's much nicer structural pictures of the same thing.
Right, and maybe we can discuss in depth later, but as a small experiment, what I was doing was, for given epitope sequences, I just took the first 4 bases, like the first 3, 4 amino acids, and first 3 and the last 3, 4 ones, and then like try to see how many unique values do I get for them, and I expected that should be much lower because for the epitopes that are actually binding to the alleles using NetMHC, and I didn't get the expected result exactly, and didn't get exactly like certain the starting region and ending region having more binding.
Let's talk about that a little later, maybe. So
Yeah.
I don't completely understand, but I don't want to
Right. Sorry,
confuse
I'm unable to explain clearly. Sorry.
Cool. So this is what I talked about. Cool. So then the as was just mentioned already. So as I said, the most selective step is this MHC class 1 molecule binding, and there's also MHC class 2 molecules. I was trying to keep it simple, so we're sticking to MHC class 1. MHC class 2 is more complicated a little bit, but it's the same basic principles. So what we have to experimentally probe which peptides can bind MHC class 1 molecules is assays such as this, which were where we have a competitive MHC binding assay. We have a peptide, and we have a labeled known high affinity ligand. And then essentially we are titrating in the peptide that we want to examine, and we're asking at which concentration do we outcompete our known labeled ligand that only half of what normally binds anymore. So you get a IC50 value, which is an inhibitory concentration 50%. So at which concentration of this peptide of interest do you outcompete 50% of this? Meaning, if you have a low inhibitory concentration, then the peptide that you're looking at is actually a high affinity binder, which always is confusing. So, so this is like a very high affinity binder where you use 3.6 nanomolar of this peptide outcompetes 50% of the reference ligand, versus this guy, you can give up to 50,000 and you still don't have any outcompetition. So this is the kind of data we have. And you get these kind of peptide and affinity value pairs And you can say, well, okay, why don't we just measure every possible peptide? And the reason, obviously, why you don't do that is because there's only finite amounts of money. There's way too many possible peptides. Every experiment costs money. And when you look at these data, you can actually learn rules instead and try to predict what is actually the outcome of such an experiment. Essentially, the goal here then is predicting for a given peptide sequence, any peptide sequence, can we say which one is, what's the affinity of that peptide? So as I was trying to say, it's impossible to measure all peptides. So we want to predict this by learning rules from the data that we have. And essentially, machine learning is a fancy term for function fitting. And ultimately, this means that you have some kind of function space. And I'm talking functions in very general. If you talk about the CNN or whatever, neural network architectures, support vector machines or whatever, ultimately, you can all think of those as a function space where you have a high dimensional set of functions that you can possibly construct with whatever algorithm you're looking at. And you can go through them and ask which one of this best reproduces, given the sequence examples that I have, the affinities that I found. And again, there's like many different approaches that do this, and there's better, or I mean, the difference between them ultimately is what do they mean by find? Definitely that has changed a lot in recent years. Then what's the function space that is being considered? And what do you consider an appropriate approximation of your found values versus the measured affinities? And the simple example that I like to show, not at all because it's the best way of doing it, but because it illustrates what machine learning in this context means, is that, okay, you have your experimental examples of experimental measured affinities, peptide sequences, and now you want to kind of summarize this data. So you can actually ask, okay, for a given peptide sequence and the measured affinity, we typically use log IC50 values, which are roughly proportional to binding free energies, that for a given peptide, I want to find a matrix where if I take the residues in the peptide, like the F in position 1, the Q in position 2, etc., I sum up these values, I want to get as close as possible to these measurements. And if I have enough peptide sequences, then I can essentially directly fit a matrix like this and ask which matrix like this minimizes the distance between the predictions that I'm getting when I use that matrix for these peptides and the measurements I have. And then you have, and essentially machine learning, such a positional scoring matrix means minimizing, as I said, the difference between the predicted values and the measured values by varying the matrix values. And then you can think of the next step of saying something, okay, I know that these are not just letters. I know that there's biological similarities between them. I'm expecting that the matrix is gonna probably have similar values for D and E and K and R, which have a similar charge versus and they are most likely going to be opposite and stuff like that. And the hydrophobic should be the same. There's a lot of things that you can add onto this kind of matrix learning principle. It ultimately stays the same though. You have examples from which you want to learn. So where do you get the examples from? That is actually one of the most crucial parts. Many of people currently are focusing on neural network architectures and things like that. And that is definitely important and an interesting area. But if you don't start with a sufficient data set that has sufficiently high quality, you cannot rescue yourself, no matter how fancy your algorithms are, no matter how fancy your computing architecture is. If the input data is small or questionable, you're not going to learn anything from it, or, well, you're going to be stuck with it. So actually the reason why I came to La Jolla was the Immunology database being founded. So I was one of these people working on these algorithms, and I was constantly struggling with getting experimentalists to give me the data that they had in order to make them better, and also to compare different algorithms out there and to really rationally learn what is better and what is worse. Because the problem typically was when I was working essentially as a PhD student in a lab in Germany where we didn't have any of our own data, we were stuck with trying to ask other people, please help us, or reading through tons and tons of papers and trying to extract the data. So I was super excited when I heard that the immune epitope database was going to come into being at the La Jolla Institute, actually led by Alex Sette, the PI at the time, which was going to go through all the literature and do a good job of cataloging all this data that's in there. And this is capturing data from allergens, infectious disease, autoimmune diseases, transplantation, alloantigens, all kinds of disease types, all kinds of hosts like humans, primates, etc. And today, now we are at over 1 million different structures that have been analyzed in over 2.5 million assays from over 21,000 papers. So the way the data comes in the database is there's actually curators reading the data and entering them essentially into a structured format. So the data in papers, it's perfectly fine. There's nothing wrong with it. But if you want to search across thousands of papers, you want to have it in a format that you can search across. So you meaning you have to agree on how to call things, because it's going to be different from paper to paper. If you go from autoimmune diseases to infectious diseases, people call essentially the same thing completely differently. Then also you have data hidden in papers, essentially by it being in figures, which that's actually the most important part. If you think about it, when you read a paper, I'm sure, at least I know I do, I scan through the figures first, because that's typically the most important part of the information. And most of the NLP kind of text mining stuff is ignoring them completely. So anyway, so the work we're doing is capturing this data into a structured format that allows us to query across different data in the scope of the IDB, which is essentially all literature involving epitopes being characterized. So the way we are getting the literature is we are querying the PubMed for new outcoming papers every 2 weeks. And then we're using a text classifier. I could have a whole different talk about that, identifying based on word frequencies and interesting combinations, what are likely documents that are relevant and epitope related. Then we do a manual abstract scan of identifying what is actually really relevant, and that then ultimately gets given to a curator. These are PhD level scientists, typically former wet lab scientists, who have intricate knowledge of the kinds of assays being run, and they are able to capture the information from the papers in a way that makes it, catalogizable, I guess. Okay, and then this is the breakdown of the figures of the data that we have right now. So over half of the data is from infectious diseases, which is what the IDB was originally funded for. This is the homepage of the IDB. So you can come here and search for any epitope that you care about. There's the search interface straight in the middle. And for example, if you want to know like what are our HCV T-cell epitopes in humans, you can go in and ask for hepatitis C, humans, and T-cell epitopes, and hit search, and you get data like this back where you have your list of epitopes. And then you can click on the detail links here and get your specific information back, where get like a specific epitope sequences and all where it has been tested in. So I wanted to briefly show that actually. Okay, now I have my thing back. Okay, so I have a question a while back. Do cells really need to make their own amino acids if they can just take ones from degraded cells? That's a good question. No, they probably. I was not necessarily saying they have to make reuse their own, but definitely they have to make new proteins. So they do have to synthesize new proteins, and essentially like tying the amino acids back together in a different kind of string, if you want. Okay, I wanted to briefly jump in here and just do a live demo, and I hope that works. Live demos are always a bad idea. So this is idb.org. It's freely accessible. So if you go in here and now let's do SARS-CoV-2, as that's new, and data that we have in humans and that are restricted by, let's do, yeah, well, actually, why don't we look at all and see what happens. So the slides I had put together on this were from the start of the year. If you had looked at the start of the year, there would have been no data at all, because SARS-CoV-2 actually just got its name kind of in March. The sequence was discovered in January. But now there's like 30 papers out here that have been defining different epitope targets in SARS-CoV-2, and you can look them up. And so specifically, apparently the targets that have been studied is the spike glycoprotein, and so the spike is the dominant target here, and then you can actually look at the distribution of the epitopes identified across the glycoprotein. You can see some regions here where you have more and less areas of reactivity. Now, I must say, with 30 papers, this is far from comprehensive, and there's papers coming out on SARS, as you probably know, pretty much every week. I wouldn't yet take this as comprehensive in any way, shape, or form, but I think it's pretty impressive how much data has been out there already in the database for a pathogen that was unknown just a few, essentially the first paper came out like 6 months ago. So yeah, but no, I think I can stop with that here. Are there any questions on this part? Structure files are notoriously a nightmare due to evolving file conventions with PDB. What are some strategies for representing structures as input for machine learning? Very good question. I don't think I have a good answer. I know we have been fighting with MMSIF and other conventions that then didn't really catch on or so. We are still stuck with the original PDB file conventions. Like we are, okay. I guess like as many tools, I'm not the structural biologist, but as I understand it, most of the stuff that we are doing is still then converting back to the ancient PDB format, which has all its own problems. It would be fantastic if somebody would be coming up with a more consistent and stable format for structural data, but that's not our that's not our fault. That's not a very useful answer, unfortunately. Yeah, but maybe I can do a different query here. So specifically, if you're interested in whatever training and machine learning method, you can say, okay, I want to have for exactly what I showed. If I want to have MHC ligand assays, actually more specifically MHC binding assays, you can find them here. So this would be MHC binding assays, specifically restricted for HLA. So one specific molecule like A star 2402, to just use something different than A021, which everybody else does. And if I search for that, let's see. So at this point, if I then look at the specific individual assays, so there would be like 5,488 data points of specific peptides restricted to HLA A2402, and then you have your binding affinities measured next to it. So that's more than enough data to try and test all kinds of machine learning methods that you can think of to test this out. So the next question here, is there research going on where we can use epitopes to predict the antibody structure sequence that will bind to that epitope or the reverse? So antibodies, okay, cool. Why don't we do that? And let's pick something other than SARS. So if we're doing antibodies, let's say to, I don't know, influenza. So this is the epitopes that have been characterized for being bound by B cell antibodies. So by B cell / antibodies, obviously tricky. In influenza A virus, you see like the 7,000 different experiments, 1,200 epitopes. Now, if I search for Has receptor sequence, this receptor here is meant to be the antibody. So these are now the paired ones where we have the specific epitope being recognized and the receptor to go with it. So these are, so we have 101 antibodies that have a known specificity. Let's go here, let's hope that works. So you have like antibodies have like a heavy chain and a light chain. This is a heavy chain with this sequence, and the light chain with this sequence. And okay, apparently here was a crystal structure, and here you have the residues it binds to. So this is all about like the data availability. So the question was, is there research going on that kind of ties to the specific antibody binding a specific epitope? So yes, there is. And this is essentially what I was trying to show here, is the data that we will be using. So here we now have not just the epitopes, but also the receptors that bind to it. And I should be removing this guy. I had been working on a number of projects like this before, and there's definitely a lot of information we can gain from the receptors. Typically, when you're doing antibody epitope predictions, the question historically was, given an antigen, given like a protein, where can an antibody bind? And typically the answer is pretty much anywhere that is accessible on the surface. And that's not a very useful answer. So the more interesting answer is like, okay, given this specific antibody, where would it bind? And definitely, I mean, essentially you're looking then from complementary interfaces, and we've been trying like traditional docking mechanisms. They don't work so well for this subject area, but there have been development of antibody-specific docking mechanisms, algorithms that are looking for the way antibodies specifically kind of mature towards binding to a certain antigen. And yes, that is somewhat working. It has been far below my expectations. I was also hoping that we would get much more of this receptor-specific epitope data. That hasn't been happening. So it used to be we had much more antibody-antigen pair data just based on crystal structures, because people crystallize antibodies bound to antigens, and that's great. But there aren't that many high throughput mechanisms where you can pull out antibodies that all bind the same epitopes. It's easier to do for T cells. And I think that's currently holding the field back. So that was a very convoluted way of saying yes, that research is ongoing, and it's still challenging. It's definitely something that we are super interested in. And I'm I'm sure it's going to work. It has to work. It should make antibody epitope prediction so much better. Okay. Okay, I think I've played around long enough with the database to give you an idea of like it is really a wealth of information that is now captured here. And one of the cool things about talking about is like different people use this for very different things. I mean, you can go in here and specifically ask for all kinds of questions, like also like what viruses infect horses or so. You can find that out based on the data that we have captured. Obviously not comprehensively, but yeah. So let me go back to the slides. Yeah, that's what I wanted to do right now, and I did. So to summarize what we had so far, so there's two main arms of adaptive immunity. There's the humoral immunity, which is like antibodies that are secreted and floating around in blood and trying to bind on the surface of pathogens that are floating around in order to stop them from infecting cells. And then you have the cellular immunity T cells, which are kind of coming in when things are too late. So as in like when you already have cells that are infected, you want to detect which cells are infected and clear that by killing them, or otherwise make the cell recognize there's something going on and kill them themselves. So adaptive immunity recognizes specific parts or epitopes of pathogens. So epitope just means it's the specific molecular site that the T cell receptor or B cell receptor will bind to. And I say here of pathogens, but the same is true for like cancer antigens or allergens. Epitope is just the word of essentially the binding site of the adaptive receptor. We can use algorithms to identify what epitopes are likely targets of adaptive immune responses. And the way to train these algorithms is to have lots of good examples. And that's what we're trying to provide in the immune epitope database, a catalog of experimentally defined immune epitopes. Okay. So now we come to the part that Alex probably covered. So I'll be going some things quickly, and then we can talk about specific areas that maybe he left out or so, but I'll still go through because not everybody has seen this yet. So as you know, SARS-CoV-2 was sequenced only in January 2020, and it was called different things. SARS-CoV-2 actually took a while to settle as the name, and to get an entry into the NCBI taxonomy, which we all rely on, took 6 months or so. So the IDB, the Immune Epitope Database, which is one of the key things that I work on, was specifically funded to speed up the ability to understand and respond to new bioterror threats. So terror in parentheses here. When this was funded, that was directly after 9/11, where there was a concern that that was an attack that the US was not prepared for, and might there not be others? And the idea of somebody releasing designed pathogen designed to kill as many people as possible was a real and realistic concern that there was no preparation for at all. And so there was an idea of let's fund the scientific infrastructure to be able to rapidly respond to if something like that were to happen. And then quickly after 9/11 terror scares kind of went down, it became clear that this is a good idea overall. We should be prepared for a new pathogen arising, and it doesn't really, that's why terror here is in parentheses. The kind of tools that you're building to rapidly analyze a newly arising pathogen are pretty much the same if that's a designed one or if it's something like SARS-CoV-2. So we have been, whenever something like this occurs, when a new pathogen comes out like SARS-CoV-2, or I mean, there have been the biggest thing for us was the pandemic flu in 2009. We've been trying to say as soon as possible, okay, what can we say about this pathogen given the data that we have in the IDB? Can we compare it to what other epitopes have we defined in other related coronaviruses? Or also, can we predict based on the tools that we've developed based on machine learning, what are the targets in this new pathogen? So, and this is actually something I'm super proud of. Like SARS-CoV-2 was sequenced in January 2020. we had our initial analysis done and published on bioRxiv in February, and then in March it was published. And at the same time with the publication, we went ahead, actually no, before that we ordered the peptides and tried to start the experimental studies to then experimentally investigate further. One of the things I really do have to point out here, the bioinformatics tools we are providing here will not, as we design them, substitute the need for experimental testing. One of the biggest advantages we have is that we can narrow down the kind of scope that you have to go through, the kind of candidates you can ignore by, okay, bullshit. So we can predict a set of peptides that is going to be covering like 95% or so of all the possible immune targets, and allow you to study that using 1% or so of the possible things you would have to study otherwise. So that is the biggest advantage of the bioinformatics, to make some things feasible that didn't used to be feasible, and then also to help interpret them. So this was the first paper, and that was really Alba Grifoni who was doing this work, who was an experimentalist in Alex's lab, and she was leading the analysis here in combination with obviously the tools team as well. So originally when the sequence came out there, it says scarce data, but there was no data available on SARS-CoV-2 when the sequence came out in terms of what the epitope targets were. The sequence comparisons that we did and many others did at the time clearly showed that SARS-CoV-2 is very different from MERS. So this is the different pathologic coronaviruses in humans, but it's quite similar to SARS-CoV-1. The last SARS pandemic, or which was much smaller, I don't think, no, it wasn't a pandemic really. So what we did was we extracted the known SARS-CoV epitopes, B cell and T cell epitopes. And then we looked at the novel SARS-CoV-2 coronavirus, and based on sequence homology, identified what things, to the things that we know from SARS coronavirus, what things are likely conserved in them. And ultimately, we found 4 regions here with overlap, significant overlap, that we thought, okay, this might be things that have been previously described to be reactive, and maybe they are reactive now again. This is for B cell epitopes. And then also we predicted and looked for essentially which are the potential targets, and we find a lot more. And sorry, the overlap here is both predicted and experimentally confirmed. We have like 4 very, very high likelihood targets here. And then for T cell epitopes, we scan for possible peptides based and predicted them. The predictions for T cell epitopes work quite well. Predicted CD4 and CD8 T cell epitopes, and identified then the overlap based on those identified based on sequence homology by T cell epitopes in SARS-CoV-1. But again, these were scarce. It doesn't say it here, but these were scarce. So the overlap approach of previously known epitopes was probably overly conservative, because not probably, we know at this point that it was extremely overly conservative, because I mean the SARS pandemic stopped quite soon after it began, so there weren't really any huge studies being done that allowed people to comprehensively study it. So anyway, so we had these sets of peptides, and then used them, specifically the peptide sets for T cell epitopes that are essentially covering, which you can just synthesize quite easily and fast, and then probe for what are the T cell responses that you now find in people who are infected or not with SARS-CoV-2. So here is where I was going to go into. Let me stop here again and ask if there are any questions. Also, actually, I've been taking more questions, which is good. Yeah, any questions here? And I can dive directly into the different papers that ultimately came out of this. I hope, I guess I was trying to say, like, so far this is covering our bioinformatics approaches. I'm hoping from I hope I gave you some background on the biology of adaptive immunity. I hope I showed you that we are cataloging epitope data in the IDB, and that we are using that to train machine learning algorithms to predict outcomes for a new pathogen, and that we've applied it to SARS-CoV-2. And I went targetedly slowly because you heard Alex talk about some of the findings before, but I'm happy to go into more details on the experimental results from this, or also on the bioinformatics background that led up to this.
So in one of the slides where you showed the matrix with different, for different amino acids at different positions. Yeah, this one. So here you showed that this is for allele A201, right? So
yeah,
do we have these matrices for like all the alleles that we know, or is there, because so some tools like NetMHC, I know that they use the fact that similar alleles or alleles with the same type will have different epitope binding profiles, and they measure the distance between the alleles and use that to predict the epitope bindings. So
yeah,
how do we?
Yeah. So
Basically, how do you account for different alleles in this analysis?
Right. So here, for a matrix-based prediction, there is a different matrix for every allele. And those are essentially individually trained. That was the original approach. For every allele, you have a set of peptides that bind with different affinities, and then you retrain your matrices. But the problem is that there's like thousands of alleles, and they vary, and many of them don't have enough experimental binding data to have like a targeted training. So essentially what NetMHC PAN does is it takes not just, this is a HLA-A0201, is essentially a protein, and then it encodes the protein itself using a pseudo sequence, using essentially the amino acids that are making up the binding pocket of this allele. And then it takes essentially all the data that you have for A0201 binding. So you have the pseudo sequence representing the molecule it binds to, the peptide that it binds, and the affinity. And then you're training one neural network that has essentially accepts any kind of pseudo sequence for the allele. And then you can include their alleles that have never been measured before. So the reason why I'm not showing that is way more complicated. Here I'm trying to show the matrices, not because I'm saying we should be using matrices, but because at least this I can show. I cannot really spell out how a neural network works easily on a slide like this.
Yeah, but actually, that's what I was thinking too. So NetMHC PAN is ANN, so actually that won't even use such matrices, right?
No,
yeah,
it'll not. No, but actually, when you deconvolute the network, it looks quite similar to this. There's actually not that much. Well, that goes quite deep into the neural network architecture things. But there's actually not much like nonlinear interactions in neural networks. The best advantage they have is really the generalization across alleles. That's been working really, really well. But the binding mode of peptides to MHC is really quite linear. So the positions are quite independent, surprisingly. I never thought that was true, but it is, apparently.
Right. But doesn't that, like the fact that we use the similar alleles for different alleles that we have never measured before, doesn't that lead to a bit loss of sensitivity at the subtype of that allele level?
So this has been a concern that, I mean, we have this approach of trying to represent the distance of the allele that we're making the prediction for to the closest where we actually data for. And we do see that if you go, the farther away you go, the lower the predictive quality gets. So that definitely is a real concern. but yeah, I mean, the best thing what we can do is measure more data for the alleles that are missing. So apart from that, it's still better than nothing.
Right, that makes sense.
Anybody else have any questions here? Okay, I see a hand up. Gautam Prakabar. Maybe that's a mistake.
Oh, sorry, I forgot to put my hand down. That was from the beginning of the talk.
Okay. Okay, we hit 6 o'clock. I don't know if there's any more questions. I'd be happy to answer all the things by emails going forward. And I guess, I mean, one of the take-home messages that we've really been getting, so since the pandemic started, we've pretty much doubled the amount of people who are using the tools, using the website, and pretty much worldwide. And also the number of citations we're getting is through the roof. Because obviously the topic that you had was like research during a pandemic. This is one of the things you can do. And it's kind of, this is one of the things that the whole project was intended for. So it's actually great from that point of view that we have been able to contribute and find things and lead to a number of publications that have been very high profile. And just like yesterday, another one came out discussed in the New York Times on longevity of the response. So it's great to see how these things are actually helping and are speeding up research. And I mean, it's not like we can say now we've cured it or anything like that. Obviously, the research enterprise is a big one, but it's awesome to play our role in it, and it's fun for us to see how very different people are using the tools and the database, yeah, to make progress towards a really important goal. And I think with that, I would stop.
Okay, thank you for giving us a talk. It was really interesting.